what we covered? No? Everybody good? Eyesight, chemoreceptors, photoreceptors. I'm sitting right here. I'm not getting up. I might lay down on the desk. Hey, can somebody do me a big favor? Because I still don't have my clicker base from the book representative. Oh, thank you, hon. Will somebody start a sign-in sheet? We got one going right there. So start you can start it and just sign in so that I can take attendance that way. Okay, so we left off talking about the nerve pathway. So this is a pathway. Gustatory receptors are located where? Quick review. Where on the tongue? In taste buds. Their little dendrites stick in that taste pore region, right? Just to recap, similar to what we saw in the nasal epithelium, we see supporting cells. We have vallate, fungiform, circumvallate. We have a couple of different types of papillae, little areas, little bumps where we find these taste buds. The taste buds look like that little clove of garlic. And we find the supporting cells holding in the receptors for your sense of taste. Again, chemoreceptors. So what do we need in order for them to be fired off? Some sort of chemicals dissolved in what? Some sort of fluid. The fluid we're talking about here is saliva. So you're going to do some tests in lab this week to prove that your gustatory receptors won't work if you don't have any saliva. You know what you're going to do? Yeah, you're going to get a piece of paper towel, dry off your tongue, and put some sugar on it. And see how long it takes you to actually taste it. It's going to take a while, right? Because you'll have to do what in order for, you'll have to start building up some saliva before you can actually taste it. Yeah. So you'll do that in lab along with some other fun, exciting things this week. So once the receptors are fired off, where does that information go? By the way, your sense of smell, where does it go for processing? I think that was one of your, better know this when you come to class on Tuesdays. All right, now that you see this diagram, take another guess. Now this is your gustatory pathway. So you have cranial nerves. Cranial nerves what?
7.
Cranial nerves 7. Facial, vagus 10. Glossopharyngeal. So you have several different cranial nerves that are going to send information for your sense of taste to what portion of your brain for processing? It's going to take a trip through that thalamic region, but where is it going to end up being processed? In the gustatory cortex. In what lobe is that? The temporal lobe, yes? So now answer my question, where are nasal information processed? In the temporal lobe as well. Who's going to bring that information for processing? Cranial nerve what? 1. What's the name of it? Olfactory. Yes? So the two are very closely linked together with respect to pathway. So a lot of what you taste is actually what you smell, because the two work together to process the information. So you're going to see that in lab today as well. We're going to block off your sense of smell and see if you can taste And you'll be surprised what you can't taste until you smell it, or until you chew it and feel something else. Get another cue, like texture. Those are some of the different cues to what identifying the foods that you have in your mouth. So, some different homeostatic imbalances talk about pathways that get fired off during different phenomena. People can have taste disorders and smell disorders, and it can be due to different diseases. Different medications can have this effect as well, which takes the pleasure out of eating when you can't smell it. Anybody know anybody or have that problem? Can't smell? And she lost her ability to, yeah.
It was interesting when Colton was a baby, taking him over there because she never knew
Oh, need to be changed? Which is probably good for her, but not good for him. All right, so they talk about some of these different disorders under homeostatic imbalances on page 570 in the text. All right, so last but not least in this chapter, hearing. Hearing and sense of balance kind of hang out in the same area in the inner ear, and the receptors share a similar cranial nerve 8, the vestibulocochlear cranial nerve 8. So when we talk about information being processed for hearing and equilibrium, we'll see that this cranial nerve and the branches of this cranial nerve are responsible for that. So on page 571, we see a much better figure. I got to redo my figures. Something happened here. Everything's all mushed up together. So we see the external structures for this particular sense. Now, can you hear without this? Yeah. What's this for?
Brings it in, right?
Yeah, it helps to channel in sound waves. What kind of sense are these two that we're going to talk about? What kind of stimulus are we talking about? It's not a chemoreceptor we're talking about. It's not a photoreceptor. It's a mechanoreceptor. It's a physical change. So when we talk about sound waves coming in and causing a physical change, we're talking about mechanoreceptors in these inner ear. So the structure of the ear is basically like a funnel. And then it's nice, it holds your glasses on, but it funnels in the sound. You can hear without it. You might not be able to hear as precisely, but you could hear just fine. It funnels in the sound. That auricle on the outside brings those sound waves to the external acoustic meatus, or external auditory canal, sometimes it's referred to. And it's going to help to channel that sound, excuse me, into the middle portion of the ear. So that's the outer ear. The middle ear is going to contain some of the smallest bones that we find in the body. 3 of the smallest bones in your body. The incus, the malleus, and the stapes are found in the middle ear. And the entrance to the middle ear is a drum. It's a piece of membranous-like tissue that's pulled across. And when sound waves come in, they hit the drum. Did you ever play a drum in the band? You've played a drum, even if you weren't in the band. Everybody's hit a drum, right? You take something hard, you hit it, and what happens to the drum head? It vibrates. So what do you think happens to your eardrum, or what's another name for your eardrum? The tympanic membrane, when sound waves hit it. It vibrates. When it vibrates, it's going to move the 3 bones behind it. When the 3 bones behind it move, they're going to set up waves of fluid. Those waves of fluid are going to be found inside the inner ear apparatus. When we talk about hearing, we're going to be talking about a little spiral thing that kind of looks like a conch shell. Do you ever go to the beach and see those shells with the little passageways. That's sort of what your cochlea looks like. Inside the cochlea is where we're going to find the receptors for hearing. So the middle ear contains those bones. The entrance to the middle ear is the tympanic membrane. And then there's a little tube that sort of offsets any pressure buildup that might occur in the middle ear. It's called the pharyngotympanic tube. It used to be called the eustachian tube. There should be a little bit of fluid in there, basically to keep all the cells moist, but there shouldn't be too much fluid in there. So fluid is constantly being produced, and then excess is being drained out that pharyngotympanic tube. Where does it go? Do you know? Yeah, down your throat, and you swallow it. You're swallowing a whole bunch of stuff from your secretions in your head. But sometimes, especially when you're little, that tube doesn't quite tilt down. Did you have a question? Yeah. So, as you develop, as your skull bones develop, as your facial bones develop, sometimes that tube doesn't quite point down for a couple years. And any of you who have had kids, you know exactly what I mean. You have ear infection, ear infection, ear infection, ear infection, and then they hit 2 or 3 years old and bam, no more ear infections. Or it keeps happening and they have to somehow equalize the pressure. Because what happens is that fluid builds up behind the tympanic membrane and can actually cause it to burst. So what they do is they put tubes in your ears. And basically what it looks like is a tiny little straw that they poke through your tympanic membrane. And that's going to allow you to equalize the pressure on either side of those chambers until that eustachian tube or pharyngotympanic tube properly angles itself. Sometimes it never will. My husband has a problem. He's way past being little, and he still has a problem with his tube. It's not angled properly. Every time he gets a sinus infection or anything nasal, he gets a problem with fluid backing up into his middle ear. Yeah.
So when you go, like I've been scuba diving multiple times, and my ears always hurt and pop. So would it be this tube that is what's hurting?
Yeah. Well, when you go up in a plane, when you equalize, what did they tell you to do? You don't have to chew the gum. All you have to do is go. But you just look stupid standing there But if you open your mouth or you yawn, you equalize the pressure there. So that's what's happening. You have to equalize the pressure inside and outside that chamber because it builds up inside, especially when you go down low or up high. Does that make sense? So, the reason the pressure in those chambers is so important is because you want the sound waves to travel. If there's too much pressure inside that chamber, it's going to be like me when you wanted to go play that drum, me putting my hand on it and say, okay, now go play the drum. What's going to happen when you hit the drum head? It's not going to vibrate properly because the pressure is too high.
So it's not everything sounds really
Exactly. So that's to equalize the pressure so sound waves can travel and do what they're supposed to do freely. So that is the middle ear. When we talk about the inner ear, and this is what we see for middle ear. Oh my God, that diagram's so messed up. I apologize. So the stapes, or stirrup, because sometimes we call them the hammer, the anvil, and the stirrup, because that's kind of what they look like. Well, not really, but use your imagination. The stirrup is sitting on another little membranous window. And when sound waves hit the tympanic membrane and move the bones, that stirrup pushes on that window. What's the name of that window? Yep, it's called the oval window. And behind the oval window, we're going to see the apparatus for hearing, the cochlea. So in the internal ear, we see this setup. The cochlea is full of fluid, and there's little chambers, just like that shell that you find on the beach, that set up little canals that have fluid inside them. In lab, when you look at your microscope of the cochlea today, I want you to pay close attention to finding what we call the spiral organ of Corti, which is basically the area where you're going to find the receptors for hearing. So imagine that shell if I flip it this way and cut it. So when you look under the microscope, you're going to go under low power and you're going to see something that looks like this. So I took that thing and I cut it. Yes? Now, what you're going to see then is this something that kind of looks like this. Maybe not in all the circles, but in some of them. What's running this way is the fluid. I hope this is making sense. So think about that little stapes when a sound comes in and hits the drum. All the little bones start to move. This little thing starts to push on that window and make waves that are going to flow through this little setup. As the waves flow through, they'll disrupt the receptors and rub against them or cause things to rub against them that we'll see in a minute. And that's what's going to fire them off for our sense of sound. So different frequencies and pitches of sound waves are actually setting up fluid waves that are allowing you to hear. So inside the cochlea, we're going to find that those little strings that I drew first, you're going to go in and magnify those under high power. And what you're going to see, and see it's much better on the diagram. My drawing was pretty crappy. But here we see that little string that comes across. And here we see the receptors blown up here for hearing. So we call it the spiral organ of Corti. And the fluid is going to flow through canals called bony labyrinth and membranous labyrinth. So we have a little hard structure and a little membranous structure that set up little canals within the cochlea. And when I make that fluid flow, did you ever see one of those little wave machines? You know, at Brooks, what's that store where they sell all kinds of, I keep saying Brooks Brothers, that's not, Brookstone. Yeah, you go to Brookstone at the mall and you see the little wave machine. That's kind of what's going on within your cochlea. So when sound waves hit the tympanic membrane and create waves within this fluid, within the bony labyrinth and membranous labyrinth, that fluid is of two types. Within the bony labyrinth, we have the perilymph, and within the membranous labyrinth, we have the endolymph. And those are the waves that are going to create sound for us. So different intensity waves are going to fire off different receptors. So sometimes I talk really loud. Yes? And sometimes I talk really soft. So just like with our sense of taste and our sense of smell, different, or sight, different wavelengths in different patterns will fire off different receptors. Different sound waves in different patterns will fire off different receptors for processing in the brain.
Like the cochlea implant, how does that work?
I don't know.
That's amazing.
Yeah.
My husband, two of his cousins have it done. They were deaf their whole lives. I guess a year.
Yep, it is. It is. It would basically take the place of these guys here. I don't know the basic principle of it, but yeah, it would take the place of these receptors, sending that information for processing in the brain. And you think about sound, and it's basically a series of different wavelengths coming in different patterns.
Yeah, they're amazing.
Mhm.
They're from a small island in Canada. First thing he did was he went to beach, and that's the first time, that's the first thing he heard was the ocean.
You could hear the ocean. Yeah, it's amazing. But yeah, so when they talk about the labyrinth, they're talking about the canals within this cochlea. So that fluid movement is going to pass through these canals and cause movement here. This little membrane here, called the tectorial membrane, is going to rub against a very similar setup to what we saw in all the other receptors. We see the receptors here. We see supporting cells holding them in. What do we call the receptors for hearing? They're called hair cells. And then their little cilia, or kind of like the dendrites of those cells, are called stereocilia. And that's what rubs against this tectorial membrane when those fluid waves cause that movement to happen. So the stimulus for hearing is a mechanical stimulus set up by those sound waves when sound hits that tympanic membrane. So do the sound waves actually penetrate through the inner ear? No. What do? They hit the drum. The drum moves the... Yeah, the drum moves the... Who? Who's behind the drum? The hammer, the anvil, and the stirrup, the three bones. And then that sets up what? That sets up those waves, okay? Those waves are what's going to fire off the receptors. That's what's going to get processed for hearing.
I think that cochlear implant just bypasses the middle ear.
Does it bypass and goes right to the inner ear? Yeah, because if you disrupt the pathway in any way, so say you had a problem with your tympanic membrane, or say you had degeneration of your, we call them the auditory ossicles, those three bones. Anything that disrupts the pathway would disrupt you from hearing. So if we can fix wherever in the pathway that disruption was, we can fix your hearing.
When the eardrum ruptures and it has the ability to heal itself, but do you hear the same?
You're going to have, while it's healing itself, you might have a problem hearing as clearly. You know, and same, back to the husband again, his eardrum would rupture on a regular basis until he got the problem fixed. So he was still able to hear, but his hearing has definitely been compromised. So he has to wear, my husband has to wear hearing aids because of all the years of ruptures in his membranes.
One of my kids has, he had ruptured when he was a little kid.
Yeah. So yeah, it can eventually create scar tissue and cause all kinds of problems.
Yeah,
So.
the scar tissue itself looks like an ear infection. Every time they look at it, they're like, oh, I see fluid. I'm like, oh, are you sure it's fluid?
Yeah.
And they say that.
And fluid doesn't necessarily mean there's an infection. It just means there's fluid. So that's one of the big problems with the little ones is that we tend to overprescribe antibiotics. Doesn't mean there's an infection just because there's fluid. You can use other methods to try and drain that fluid. So a lot of different homeopathic methods encourage that rather than giving antibiotics right off the bat. Because what happens when we give antibiotics too frequently? We eventually build up a tolerance to them, and we develop all of these wonderful resistant bacteria that are wreak havoc and heat and discontent through the land. So yeah, don't be quick to take antibiotics for things. Sometimes it can be viral in nature. And what are antibiotics going to do for a virus? Squat. Most people, when they get sick, do not get sick from bacterial infections. But what's the first thing the doctor will do? He'll prescribe an antibiotic. It's like, you haven't even seen me. How do you know I don't have a bacterial
I do a
infection?
lot with older people too. Like my mom is like.
Well, because they think antibiotics will save the world. Yeah. Gotta go on antibiotics if I'm sick. Well, most of the time you're not sick because of bacteria.
You know, I left my son's doctor and put fluid in his ear, and the doctor said, no, we're not going to prescribe him anything. The body will take care of it himself. Sure enough, 2 days ago.
Yeah, sometimes warm compresses. Sometimes like a certain way you can massage the neck to help drain that fluid out is the best way to do it. And then they have like topical things for pain, because the pressure does cause some pain. So you're better off doing things like that rather than taking a course of antibiotics. So, the entrance to this wonderful cavern, and that's pretty cool. I love this. We never see anything like this in our lab, but this is a cool, what do we call it? I can't think straight today, guys. The fancy microscopes that electron microscopes, thank you very much. That's what this is. So you can see the actual little stereocilia. So sound, they talk a little bit about sound, and we're going to talk a little bit about that in lab this week as well. Different sound waves produce mechanical stimulus that allows us to hear. And they talk a little bit about that on page 575 in your textbook. So sound propagation gets sent through that pathway. So when we talk about physiology of hearing, we're basically talking about the pathway I just described. Before I go too far, the entrance here is not just for hearing, but we're also going to talk a little bit about balance and equilibrium. That's these guys up here. These are called semicircular canals. And this region here, the entrance to this inner ear that's responsible for both hearing and equilibrium, is called the vestibule. These little purple things, these are the beginnings of areas, receptors for equilibrium. The receptors for hearing are going to be inside that cochlea. So, sound. There's a nice little diagram on page 576 that shows us the different types of sound waves we might come in contact with. So frequency is sound waves that are coming faster. So if we have a high frequency of sound wave, that gives us the pitch. So when somebody has a very high pitch versus a very low pitch, low frequency waves will give us that low pitch. Males tend to put off more low frequency waves. Females tend to put off what? More high frequency waves. When I want to be very loud versus be very soft, that talks about the amplitude of that wave. So different patterns, amplitude and frequency, are what's going to fire off different receptors in different patterns for us to hear. Just like we had light being bounced off an object in different patterns, firing off different receptors for us to see different patterns with vision. Am I making sense? I hope. So, just an overview then of sound, amplitude, frequency, understanding what the two mean. And we can only hear in a certain range, just like we can only see in a certain range. You ever see one of those dog whistles? Can you hear that? No. Can your dog hear it? Yeah. So just like sight, we only have receptors that will pick up a certain range. We go between 1,500 and 4,000 hertz. That's our range. Human hearing is from, wait a minute, sorry. 20 to 20,000, isn't it?
For hertz,
Yeah.
for decibels.
It's on page 576.
Oh, like that.
Yeah. 1,500 to 4,000 hertz. So, as we get older, what might happen? Yeah, these receptors might become damaged. If I over-abuse them, too many rock concerts when you were young, radio up too loud, or you work, or you work in a place where you're constantly exposed to a certain wavelength. Same husband used to work with engines. So he can no longer, it's so funny, I think he does it on purpose, but maybe he doesn't. He can't hear the buzzer when the dryer goes. He can't hear that though. I think he can, he's just lying. But certain sound waves, certain frequencies you may lose. There's a thing my daughter brought home on her iPhone. The mosquito tone, is that right? Do you have that? All right. I can't hear it. They laugh. The kids laugh at me. But why can't I hear it? Because as I get older, you lose certain places in the frequency or ability to hear certain frequencies as you age.
Middle school and high school, that's all like,
Yeah, because the teacher couldn't hear it, right? And
they'd start laughing.
they'd start giggling.
Seriously, that's just really
Yeah, yeah, stop it. So, yeah, I can't hear it.
So are the same sounds that are like annoying to me annoying to everyone else, like Neil's and Chuck?
Not necessarily, no. That's kind of like a preference thing, you know. Certain music is annoying to me and it's not to somebody else. But as far as the ability to actually hear them is what I'm talking about. So as you age, you will lose certain ranges in the frequencies that you're able to hear. And then if you abuse your hearing with, you know, not using ear protection when you mow the lawn and stuff like that, you can lose even more at a faster rate. So wear your ear protection and don't put your music up too loud. Although I do that in the car, I can't help it. So this is basically what I described to you earlier. This is the pathway that sound takes. This would make an awesome essay question on your next exam. Figure 15.30, page 577, pathway of sound waves and resonance of the basilar membrane. So again, those waves are going to be passed in a pattern through the cochlea, and that fluid passing through that what we call spiral organ of Corti, where those receptors are found, are what's going to fire those receptors off, and then we're going to process that information in the temporal region as well. The receptors, stereocilia, are here. Pretty cool. And where do we send that information for processing? Now we have a cranial nerve that's responsible for both of these guys, equilibrium and hearing. It's called the vestibulocochlear cranial nerve 8. So guess what branch of that nerve is sending information for hearing to your brain for processing? It's got two names, right? Two parts to its name. Right, the cochlear branch of that nerve is what we're looking at here. So we send information for processing, again, in those temporal regions, Broca's region, all that stuff we learned last semester is being sent from the spiral organ of Corti in the cochlea down the vestibulocochlear cranial nerve, 8 for processing in the brain. Okay? So we have the ability to process information about pitch and amplitude and distance and direction based on the way this information comes in. So we'll do a few little tests in lab with one of those little tick-tick clocks. That'll help us see how we can localize sound in different places. And that's what they talk about in our textbook. Now the other group of receptors in this area can be found to help with balance and equilibrium. We find these guys in that vestibule region, in an area called the macula of the utricle and the macula of the saccule. And the structure is very similar to what we saw in the spiral organ of Corti. We have the same kind of setup with all these guys, some kind of supporting cell holding up some kind of receptor. The receptors here again are called hair cells, and the little dendrites that stick up again are called stereocilia. But in this case, they're going to stick up and sort of embed themselves in that membrane that's going to cause change. That membrane is called the otolithic membrane. And inside the membrane, there's these little crystalline structures called otoliths, or ear stones. Now, these are found in that vestibule area in the you can see them right here. So they're at the base of these semicircular canals. So watch what happens. Is the fluid movement going to be caused from me talking to you and little bones moving and fluid movement? Is that what's going to cause the fluid movement for? No. What's going to cause it? Do you know? Right. So when I move, or my body moves in different directions, that's what's going to set up the fluid movement in the semicircular canals. So we see we have 3 of them. One that's going to allow for movement this way, and this way. Okay? Yes?
So are your ear stones, when you have vertigo, do you have like stones in the ear and when it gets into place, like it causes vertigo?
Well, you can get overexcitation of some of these receptors is a result. So it could be a problem with the ear stones, overproduction or misplacement.
Really bad vertigo, and the physical therapist that I was working with just like held her head and just kind of moved it, and he said that it just like moved the fluid back into her ear and in a position dropped, and it took the vertigo away.
Yep. So it's whatever, if she's got fluid imbalances in those semicircular canals can cause problems with that, and overproduction, or some patients have too many ear stones for some reason during certain periods.
Knock the stones like out of place.
Yeah. Yeah. And then getting them back, it's not so easy sometimes. Yeah.
It was crazy. Her eyes, I've never seen it so bad. They were literally going side to side.
Yeah, nystagmus. And you'll do some little tests in lab too today. You did some last week, I think, with eye movement. But yeah, it can cause all kinds of problems with balance. Sometimes when you're sick and you have overproduction of fluid in the sinus passages, that can also cause problems with balance too. So we have, instead of the tectorial membrane, we have the otolithic membrane. We have these little otoliths, or ear stones. The receptors are very similar, and they're going to send information for processing in the brain, again, in that same temporal region. So the anatomy of the macula is what we're looking at when we talk about the receptors for your sense of equilibrium. They talk about a whole bunch of other things and get a little carried away. They talk about nystagmus, and that's that out of balance impulse we were just discussing.
So when the cop gives you that test, you can tell them that you have an ear infection or something. So it's
That's what I just passed him on a note. No, officer, I'm not supposed to say my ear stones
No, I don't think so. But yeah, some certain drugs or alcohol can cause your eye movement to be, and that's why they do this look at my finger thing. A couple of reasons, not only for eye movement, and yeah, you can't control it, so don't even try.
She talks from experience. You got that finger movement down really good.
No, I'm a mother, I do that all the time. And the other thing they look at is what? They look at the pupils. So certain drugs or alcohol can cause problems with you being able to dilate and constrict your pupils as well. So those are the two things they look at real quick. And they can pretty much tell right away. And then they make you do the other stuff because it's funny, and it's funny for them to watch you trip around and fall. So, yep,
do all three tests.
that's right. So that's what they describe under the macula, and this diagram is kind of neat because it shows you what's happening to all the different receptors in different head positions. You get used to certain movements, rotational movements. Some of us get dizzy from this.
I wondered why they would get dizzy from that.
Yep, well, they did in the beginning, but you can actually train yourself to be able to do that. And yes, it's a game where people. Have you seen that? You know, yeah, twirl. Yeah, isn't it awesome? A little sit and spin, wonderful.
But now if I do it immediately.
Oh yeah, now what happens? Yeah, yeah. I'm a little psychotic. I do enjoy a good carnival ride. So yeah. But that twirling, yeah, you're taught to say it.
I used to be a dancer, and we used to do our pirouettes or whatever, twirls. My dance instructor told me to pick a point on the wall, and every time I did a full circle, I would have to whip my head around and look exactly at that point, and I would not get dizzy. So
Yep.
that's how come ballerinas and ice skaters, figure skaters, can spin a million times in a circle, and then they can go and do a huge jump, and they're not dizzy. So
Yep, because they're
staring at a specific point.
Yep. So they're trained to do that. It takes time to learn to train to do that, but that's what they're talking about with respect to rotation and what happens to the different structures in those regions. So again, they're going to fluid movements from rotation can cause receptors to fire off. That's going to send a bunch of different messages in conjunction with equilibrium. So it's not just one specific place that gets information. Processing is sent out brainstem with information that's coming from a whole bunch of different places. Cerebellum's going to get involved with this whole deal as well. And visual cues are also responsible for balance. So I think, did I mention this to you? I got freaked out by the fire escape. Yeah. That visual cue was part of the information I needed for balance. Sometimes when you're looking at something that's, just off balance, like a step or something, those are visual cues that you're going to use as well for balance. So somatic receptors, pressure and tension on different joints, that's also going to be part of the message with respect to balance as well. So the vestibular receptors are only one part. You also have to incorporate all those other guys into coordination and balance. That's why that visual cue for the spinning lady helped to slow down the response, that dizzy response. Motion sickness. Anybody get motion sick? Anybody? I used to.
Yeah, if I'm not
Until I started to drive.
Yeah, if I'm not driving, I get dizzy.
Yeah, why is it different?
I don't know.
Because what do you,
Why is it that I can go on roller coasters, but I can't spin in my chair, and I can't sit in the car? What's
because it has a lot to do with some of the visual cues you see. You're overwhelmed by what you're taking in visually. Like when a person sits in a car that gets cut, this is how I got described it once, and I thought it was perfect. Normal person sits in a car, looks out the window, sees a bunch of trees, right? Person who gets motion sick sees tree, 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 tree. Yes? You see the difference? Because they're overwhelmed with the visual stimulus, whereas somebody that doesn't have that problem takes it in as a whole. Right.
I have to like block the trees off. So
Yep.
I see them like flying by me.
Exactly.
I have to, like, block them.
And when you're driving, what are you doing? You're concentrating on the road. You're not looking at all the trees, so that's why you're okay.
That's why people say they need to sit in front.
Yeah. We'll tell them just to close their eyes and sit in the back seat. Take a nappy, you'll be fine. Because it's the visual cues that's the problem for that person, and it causes them to be get sick. All right, so homeostatic imbalances then. Deafness, we have conduction deafness, sensorineural deafness. Again, in lab, you'll do a couple of different tests to see some of the differences. We can feel vibrations. So there's different types of disruption with respect to hearing. At any point, if I create a blockage, I'm going to create a problem with processing. Deaf people can feel the vibrations in music and actually feel the music. Why do I sound very different to me than I do to all the rest of you?
Do you have a blockage right in your stomach?
No. Do you ever hear a recording of yourself? You're like, what? I don't sound like that. Why do I sound different to myself than the rest of you? Because not only am I getting the sound waves I'm creating, and they're bouncing off of you and coming back, but I'm vibrating inside my own head, right? So I'm processing something very different than what you are.
You sound good to yourself.
I sound great to myself until I hear myself on a gun. Oh, God, I sound.
I have a lady that I take care of that has empty, and she can't hear herself talk. Like, she thinks she sounds normal, but she talks wicked, like, slurred, and you have to, like, really focus on what she's saying, or you can't understand her.
Yeah.
And I showed her a recording of herself one time because she wanted me to, and she got really freaked out by how the way that she sounded because she didn't.
Because she can, well, and she probably knows what she's trying to say, too,
Yeah.
so.
It's harder for her to get it across.
But yeah, that vibration in your head, you're hearing that as well. So you'll do some tests in lab that talk about differences in different types of hearing. You know, you'll put those little tuning forks on the top of your head and in the back. So you'll test for that as well. Developmental aspects, don't worry about it. We're done with chapter 15.
So, what, I'm having a hard time with this chapter. What do we really have to focus on? Just everything in here, or do we enjoy so quickly? Are we really going to be in depth?
I thought I hit everything, didn't I? I did. The only place I'm not going to get too crazy is the chemistry of vision, which goes very crazy. We kind of narrowed that one down. All right, don't go away. We're going to do chapter 16. I don't know, where'd the sign-in sheet go? All right. You want to do? I don't have my clicker base yet, fortunately. But you want to do a couple of clicker questions? No, you don't have to use it. I can't. Yeah. But we can do it in our heads. All right. Of the following senses, which accounts for the majority of sensory receptors in our body? I think it's C. It's C. Vision. Which of the following terms is a synonym for an eyelid? B, palpebrae. Pink eye. Infection of what? The conjunctiva. In order to turn the eye straight, oh, don't worry, we didn't do the muscles. That's the muscles. Cornea is actually part of what layer?
Sclera. D. Isn't it? Fibrous?
A, fibrous. What's fibrous? What are the parts of the fibrous tunic?
Sclera.
Sclera and the cornea. So the cornea is not part of the sclera, right? It's part of the fibrous outer layer. And that the cornea is the clear portion, and the sclera is the white. One more. Sympathetic fibers innervating the iris of the eye cause which response? What happens when you get What happens when you get scared? Your pupils go whoa. They get big. Yes? Did you? I think you did that last week, didn't you? No? Anyway, it's dilation. So think sympathetic, fight or flight. Somebody scares you, you're going to dilate. All right. Questions on chapter 15? Yes. So when I leave today, I will go and put your quiz up online for chapter 15.
Is it going to be very general since it's very
Always. Always.
Our first exam is 6. About.
Hopefully. So we don't get behind. I think we're pretty good.
The HESI this weekend.
Oh.
So normally after I have a class, I study this, and I know my problems with math.
It's not going to happen, right? Next week. Okay, so endocrine system. What is the endocrine system? I don't know why that's in there. So what's the endocrine system? Glands that secrete chemicals that do what? Regulate what? How? Now the nervous system and the endocrine system kind of work hand in hand. So you said regulate the body. Yes. How? What do hormones do for me? Drive me crazy. They're going to change what my cells do for a living. They're going to regulate cellular function. And that's how they're going to control different body functions. So hormones are chemicals that are going to regulate what cells do, in turn regulating body function. So like the nervous system, when I see, I fire off these receptors, I process information, I get some information for me so I can react to that information in my environment. Hormones secreted by the endocrine system are going to help more evenly control change within the body. So for example, did you guys eat breakfast this morning? Yep. You got to eat breakfast. I'm telling you, especially when you get older, eat your breakfast, eat your breakfast. If you don't eat it, drink it. And no margaritas. But like that stuff, yeah? That's good. Because, yeah. See, it's a foggy mind. But what's inside that thing? Share with the class. Yeah, you have one too. And what's that going to help her do this morning? Focus on what's going on in class, right? To help keep your what? level?
Blood sugar.
Blood sugar. Why do you need sugar in your blood? What's it for? Energy. Your cells need to make energy so they can undergo metabolism. So I need a certain amount of glucose floating around in my circulatory system at all times, so I ensure all my cells get the glucose they need to make the energy they need to function. Who's in charge of keeping track of all of that? Well, no, actually the nervous system. Okay? But the endocrine system's going to help out. So the nervous system's going to, you didn't eat your breakfast this morning. You guys did, so I'm not pointing at you. So who didn't eat their breakfast this morning? So you're going to die. Well, no, you're not going to die, because the nervous system says, Slacker didn't eat his breakfast this morning. I still need glucose.
So now his body has to work extra hard, right?
You're right. But the endocrine system, you ate last night, well, that bully for you. You used all that up. But good thing you stored some stuff, because last night when you ate, you stored some glucose in the form of glycogen in your liver, right? Okay, good. The endocrine system is going to get a message from the nervous system. Blood glucose is going down. You need to help me out here, because he didn't eat breakfast. So, pancreas is going to get the message. Blood glucose levels are going down. A hormone is going to get secreted. It's called glucagon. Glucagon is going to work its way into the circulatory system and help something change cellularly. It's going to cause cells to break down the glycogen you stored last night and free it into the circulatory system as glucose. What's that going to do to blood glucose levels? Bring them up. Okay, that's how we maintain homeostasis with the help of the endocrine system. So we have to maintain these constant levels of different things like oxygen and glucose and calcium in our blood supply at all times. So the nervous system is going to work in conjunction with the endocrine system to help do that. So the endocrine system is a series of glandular material we see in this diagram here, which is on page 592 in your textbook, that are going to secrete different chemicals to help regulate body functions to help maintain homeostasis. So the endocrine system and its hormones and the players that are involved in the endocrine system regulate everything from reproduction to growth and development. They help to maintain electrolyte, water, nutrient balance in blood, regulate cellular metabolism, energy balance, and help mobilize body defenses. So all of these hormones and all of the different players in the endocrine system are going to help with all of these different bodily functions. Within the endocrine system, you can have two different types of glands. You can have a gland that secretes its product through a tube that goes somewhere else. What kind of gland is that? That's an exocrine gland. And then a lot of the guys we're going to talk about in this chapter secrete their product right outside themselves, which is then carried through the circulatory system throughout the body. What kind of gland is that? That's my endocrine gland. So endocrinology is the study of these different organs and these different glands and the hormones and their different functions throughout the body. So we have in this chapter a lot of information, but here's how I want you to study this chapter. First of all, we have to know where all of these guys are, and that's this diagram right here. We have the pituitary gland, we have something in the neck, a couple in the neck, thyroid, parathyroid, and we have the thymus, we have the adrenals, we have the pancreas, we have some of our reproductive organs that double for the endocrine function as well. So know basically where these hormones are produced. Then we have to know the hormone and its target. Now, what do I mean by target? Yeah, what cells it affects. Does every single hormone that we're going to talk about affect every single cell? No. Only cells that have what for that hormone on their surface? Receptors. So, different hormones affect different targets in different ways. So the other thing we have to understand is how these hormones actually cause change to happen within the cell. So the next part of the book talks about the chemistry of hormones. Now, two different kinds of hormones. We can have hormones that are amino acid water-based hormones, I call them. And then we have lipid-based hormones, steroid-like hormones. Now, water-soluble versus lipid-soluble chemicals are going to act differently. If I put a lipid-soluble substance into water, what happens to it? Whoosh. It's hydrophobic. So when I talk about a lipid-soluble compound, I have to talk about a couple of other steps in order to get it to where it needs to go. I need some sort of carrier, and usually that's protein in nature, to get it where it needs to go. And we're going to see that water-soluble versus lipid-soluble hormones will react differently within the cell as well. Remember that little second messenger system that we talked about? When did we just talk about it? Yeah, when we talked about sight, right? Well, now I'm going to talk about it in detail. So water-soluble hormones create or function with second messenger systems. They don't actually get into the cell to cause change. They cause it from the outside. So the hormone in this case is what we call the first messenger. And we kind of talked about this first semester. It's going to come in contact with a receptor for that hormone. So remember, it has its own special receptor, its own special protein on the surface of cells. When it comes in contact with the receptor, it's going to activate a G protein because of the change in shape that occurs when the hormone comes in contact with the receptor. G protein is going to get on the move and come in contact with some kind of enzyme. In this particular second messenger system, the enzyme is called adenylate cyclase. There's several different kinds of second messenger systems. This one is called the cyclic AMP second messenger system. Because the second messenger that we create is called what? Cyclic AMP. So the first messenger in this second messenger system is the hormone that comes in contact with the receptor. The second messenger that's created is going to be what? Cyclic AMP. The presence of that cyclic AMP is going to cause changes chemically within the cell. It's going to activate inactive proteins when they come in contact with that second messenger. So cyclic AMP is going to activate what we call protein kinases. So there's going to be a different chemical within the cell, and it's going to cause the cell to do something different. Now, there's a whole bunch of different second messenger systems, but these are some examples of what might happen to a cell once it becomes activated by a second messenger system. So it can cause certain enzymes to be activated, or it can cause the cell to secrete certain things, or it can open up ion channels. And we know what happens when ion channels open up. They can cause changes within the cell, like muscle contraction, for example. So this second messenger system, the cyclic AMP second messenger system, is an example of how a water-soluble hormone would work. That would make an awesome essay question on your next exam. Now, in the textbook, they also talk about a couple of other second messenger systems. There's one called PIP. There's one called calcium. But this is the one I'm going to concentrate on more with respect to you understanding what a second messenger system is. Only because they give us a nice diagram. I think they give us, no, they don't give us another one. So they also describe PIP2, which is another type of second messenger system. In different cells, it creates a different second messenger. Calcium can also be another second messenger system. But this is the one I'm going to concentrate on. So if I'm not a water-soluble hormone, what other kind of hormone could I be? Could be a lipid-soluble hormone. Now, lipid-soluble hormones are going to work a little bit differently. They actually are going to enter the cell. Remember what the cell membrane was made of? Very good. Phospholipid bilayer. Remember that? So lipids are okay. They can kind of, hang in that membrane. Not too much trouble. They need some sort of receptor or carrier to make it through the fluid to the cell. And they form a complex, receptor hormone complex, once they've come into the cell. They're actually going to enter the cell this time and cause their change to occur within the nucleus. This receptor hormone complex is actually going to enter through those little nuclear pores that we talked about when we talked about protein synthesis way back when. And they can bind to a very specific place on the nucleus. They're going to cause changes by causing the cell to make different what? Proteins. Remember the second messenger system in the water-soluble hormone was the thing that caused different proteins to occur? But this binding process is actually what's going to cause different proteins to be produced in the cell when we talk about lipid-soluble hormones. So lipid-soluble hormones are going to enter the nucleus, bind to a receptor binding region there, and change the protein that is produced by the cell.
So it's like the DNA something.
Yeah, it's gonna
Avoid the ribosomes.
right. It's actually going to cause its effect right at the DNA. So it's going to affect, because of that, what is transcribed and translated from the DNA. So that's where the new protein comes on board. We're going to actually cause a new protein to be made rather than what I was making before. When that receptor complex goes away, I'll make the old proteins I made before. Remember, these proteins will then cause something different to happen within the cell. Just like we saw with the water-soluble hormones. So it could cause the cell, depending on which one it was, to secrete something or to make some enzymes and speed up reactions somehow. It can be a series of different things depending on the hormone. Did you have a question?
Well, I was trying to apply it to something that I would understand better. I don't know, sometimes they prescribe steroids if you have a rash or something. Is that
Right, to decrease inflammation.
Yeah, is that kind of how this is working?
Yeah, so it would slow down response in certain cells, inflammatory response in certain cells. So certain cells give off histamines, for example, that cause inflammation. But if I can tell cells to stop making those histamines and releasing those histamines, then I can stop the inflammation. That makes sense?
Yes.
Everybody with me? So one thing we have to understand in this chapter is the difference between the mechanism of action of hormones that are water-soluble versus lipid-soluble. Now, the next thing we have to understand is different things can cause different hormones to be released. So, the nervous system can actually stimulate, and we talked about this when we talked nervous system 1,000 times. How did the nervous system communicate with your body? It either talked to another neuron, or it talked to a muscle, or it talked to who? A gland. So the nervous system can stimulate a gland in the endocrine system to secrete a hormone. We just talked about that, right? You didn't eat your breakfast. We need some glucagon. The nervous system is going to communicate with the pancreas. The pancreas is going to secrete a hormone. The hormone's going to break down your glycogen into glucose. That's going to increase your, yes? Yep. So the nervous system or neural stimulus is one way to secrete hormones. Humoral stimulus. What's humors? What are humors? Anybody? They're actually fluids. So concentrations of different substances in your humors or fluids can actually signal the release of hormones. So the stimulus for humoral stimulus in that first example is a low concentration of calcium in capillary blood. When calcium levels start to go down, what do I have to do? Somehow I have to get more calcium into my blood. Where's the best place to meet? Bone. Thank you. I couldn't even finish the sentence. The best place to get calcium from is where I store a lot of my calcium. Bone, right? Okay. Well, I remember when we talked about bone. How do I get calcium from bone? I have to tell some cell to go break down bone matrix, right? What was the name of that cell? Osteoclasts. Osteoclasts actually used to be monocytes hanging around in the circulatory system taking a ride. Did you know that? Yeah. So they're monocytes. We're going to talk about them when we talk about blood. Until the parathyroid gets a signal, low calcium levels. We secrete a hormone called parathyroid hormone. When that hormone gets into the circulatory system and comes in contact with those monocytes just kind of hanging out for the ride, it's going to cause the monocyte to be doing something different. What's the monocyte going to change into? An osteoclast. And where is it going to go? It's going to go to the bone matrix and break it down, release calcium, and do what? Increase calcium levels in blood. So that's another way, humoral stimulus, monitoring some of those different humors or fluids for different levels. And that can also stimulate the endocrine system to release its product. And then the other way the endocrine system can release its product is get told by other hormones from other endocrine glands. So hormonal stimulus, hormones being released by the release of other hormones, can also occur as well. So it's time during my cycle, we're going to learn this last chapter, or near the last chapter this semester, we talk about the reproductive system. It's time for me to make some reproductive cells. Who's the one that's going to tell me to start maturing those cells to make reproductive cells? Another member of the endocrine system. So we have the anterior pituitary gland, which we're going to talk about in a minute, that makes a whole bunch of different hormones that are going to help stimulate other endocrine glands to secrete hormones as well. Follicle stimulating hormone, luteinizing hormone, gonadotropic releasing hormone. These are all hormones that are secreted by the pituitary gland to tell those other endocrine glands to secrete their hormones. So we'll talk about those guys in a minute. So 3 different ways a gland can secrete its product. Different humors, humoral stimulus. The nervous system can do it directly, or other hormones can do it as well. So what we're going to do next, and what you can work on before we come to class next, is I want you to make a little study chart. And on the study chart, you're going to have the gland, you're going to have the hormone, and you're going to have the target. And that's how you're going to study for this chapter, okay? And what you see in your book is there's a whole bunch of nice little tables. The first one we're going to bump into is on page 602 and 603. So when we talk about the pituitary gland, we're going to talk about 2, anterior, posterior. Anterior pituitary gland. What hormones does it make? Da-da-da-da-da-da-da. What does it affect? Da-da-da-da-da-da-da. That's how you're going to study for this chapter. So make yourself this little chart, work on it now, and when you study for this chapter, that's what you're going to study. Okay? So make sure you give me the sign-in sheet